Vincent Moulton, who's at the University of East Anglia and has been advising us on the construction of this program. So thank you very much, Vincent.
Thanks very much,
over to you.
Yeah, thanks very much. Yeah, so it's nice to be here and be able to chair the last session of today. So our first speaker is going to be Rodrigo Mendez, and you're an agronomist, I believe.
Yes.
And working in microbial ecology, and he's based at Embrapa. Is that correct?
Yeah, it's Embrapa.
Embrapa,
It's
in Brazil.
Yeah.
Good. And you're going to be telling us a little bit about the living layer. So thanks very much.
Thank you. Good afternoon, everyone. I just would like to tell where I came from. It's Embrapa. It's the translation is something like Brazilian Agricultural Research Corporation. And I work as a researcher at Embrapa, and I teach at University of São Paulo in Brazil. So this is the title of my talk that I wrote my abstract, but actually the actual title of my talk is going to be in the bottom one, the rhizosphere microbiome under fungal attack. They will explain you why. So if one of the objectives of this program is to feel out of your comfort zone, I think I'm in the right place. I'm feeling nicely uncomfortable. But let's go. So with this introduction, I'd like to give you an idea about the rationality behind all the story I'm going to tell you this afternoon. We think, and when you look at an organism, we see not only the organism, but many other genomes associated to them. And this is what I'm calling as a living layer. So between superior organisms in the environment, there is thousands and thousands of microbes doing something that we didn't know about that some years ago, but now we know. If you look at the concept of microbiome, first said by Ladenburg, is the totality of microbes, their genomes, and their interactions in a particular environment. And some people have referred to microbiome as the second genome. So now we have the view of superior organism. Nobody is alone. So if you wanted to understand the physiology or a given phenotype, we have to take into account not only the host genome, but also all other genomes associated to this genome. And in humans, we have many really nice examples. We are completely covered by different microbes, and every spot in our body has a different microbial structure. We are in and on completely filled with microbes. But what I would like to highlight, and the lesson from this study, is that the human microbiome significantly contributes to the human metabolism, providing traits that humans did not need to evolve on their own. The message here is that an organism can rely for some specific traits in the microbiome and doesn't need to have in their own. And this is the same for plants. These two papers on Nature in 2012 show that soil type and host genes determine bacterial communities in the plant root microbiome. So if you know that plant genes are able to somehow shape the microbiome associated to them, if we know how these genes work and if we can manipulate them, we can take advantage of that. So this is the bottom line. Microbes play a pivotal role in the functioning of their hosts by influencing their physiology and development. And then we move to this system here. So then the title is The Rhizosphere Microbiome Under Fungal Attack. What we want to understand is what is going on with the plant microbiome in the rhizosphere And the rhizosphere, by definition, is the portion of soil attached to the root systems of plants. This soil, called rhizosphere, is strongly affected by the plant exudates. And they have a really different microbial community when we compare with we call bulk soil. The rhizosphere is special. So when a soil-borne pathogen, in this case Rhizoctonia solani, is growing through the soil and infecting the plant, a susceptible host, how the plant can use the microbiome to defend itself. This is the goal of our study. Then let's go through these three main elements in the story. The first one is the pathogen. It's a fungus, rhizoc. This is just a YouTube video to illustrate what is going on. These are some structures of a rhizoc in the soil. They can stay in the soil for long periods, and then suddenly they start to germinate, triggered by plant exudates. The plant exudates some signals and compounds which is perceived by this fungus, and then this fungus goes growing through the soil, reaching the root systems, growing on the root systems, and they form this type of structures to infect the host plant, susceptible ones, and then infecting the plant roots, releasing an arsenal of many different lytic enzymes, destroying the plant tissue and leading the plant to death. There are two, in this case, there are two stages when the plant can be attacked: before emergence in the soil, and then you see the black lesions in the root systems and the plant is gone, or just after the emergence of the plant. The plant is attacked by the fungus in the root system and then is killed. And then we have the plant, a susceptible host. In this case, it's a sugar beet. And we have the rhizosphere microbiome. We always hear that the soil is quite complex. And with this figure, I tried to illustrate this complexity. In this figure, this side, there are many different organisms inhabiting the rhizosphere. Protozoa, virus, algae, bacteria, nematodes, fungi, archaea. And I tried to add it here, the number of genes and the size of genomes interacting to each other. It's really complex. And this is who are there. We have this zoo interacting with the plant root systems. And when you look to the microbiome, what are they doing? So if you look at the good and the bad ones, they are very well known microorganisms. The good ones are those that we know that can promote plant growth, or they can produce antibiotics to protect plants against disease, or they are able to fix nitrogen and provide nutrient to the plants. In the spectrum of this diversity of microbes interacting with the plants, in one extreme we have the good ones, and those we know because we are able to cultivate them, to grow them in the lab, and we know them. On the other side of the this spectrum, we have the bad ones. They are important because they kill plants, they cause disease in plants, and we also traditionally know them. But in between, we have this a gray area here with a massive amount of microorganisms that we can call dark matter, if you want, that we have to explore and to understand. So in the past years, when you look at the pathosystems in plants, we look at the pathogen and the plant. Now we are trying to take into account the whole microbiome together. And we are using metagenomics to try to elucidate how all these complex communities are interacting with the plant. And the system in which we use it to do these experiments, it's called suppressive soil. Suppressive soil are specific soils in which, despite of the presence of a given pathogen and a susceptible host, there is no disease for some reason. The opposite of suppressive soil is conducive soil. So we have these two types of soil, suppressive and conducive. And we have sugar beet growing in them. And then we add the pathogen. In the conducive soil, the fungus is able to infect and kill the plants. In the suppressive soil, for some reason, there is no disease, or the severity of the disease is very low. And we demonstrated that doing this type of experiment here. So here, we are measuring the disease incidence. In the suppressive soil, we have a very low disease. The opposite of suppressive soil is the conducive soil, and we have this level of disease. If we wanna demonstrate that the suppression, the nature of disease suppression is due to microorganisms, there is a way to show that. We can use, for example, heat treatment. If we take this suppressive soil without disease, and then we heat treat it for 1 hour at 80 degrees Celsius, the suppressive soil became a conducive soil because we kill the microbes. And then we can do that using temperature or using radiation, for example. If we use 50 degrees instead of 80 degrees, it's a less aggressive treatment. We have an intermediate phenotype here. And another way to illustrate the microbial nature of this phenomenon is doing the transplanting. If you take the conducive soil plus suppressive soil in a mixture of 10%, we are able to restore some level of disease suppression. This is very comparable with what people do with humans, with people diseased with gut disorders, diarrhea, and then if you want to treat this person, you can use antibiotic to clean up the gut system. Here we use temperature. And then if you want to treat this person, you can collect microbes from a healthy person and transfer to a diseased person. And the microbiome will be restored. But it's much easier work with soil and plants than feces transferring.
How long do you take to kill the plant?
Sorry?
How long you take after you kill the bacteria? How long the plant will die?
The infection, this is a dumping off, as we call. The infection in the base of the stem, it's just after 5 days after inoculation. It's just in the beginning of plant development. It's the dumping off disease. It's really quick. So understanding this system, we can think about what the plant is thinking about that. What it's the questions we have in mind. To what extent do microbes in the rhizosphere protect the plant against the pathogen? Which are the players and underlying mechanisms involved in this process? Do pathogen invasion affect microbial diversity and activity? And how? Which factor or processes determine the transition from health to disease? So is that true that the plant is really able to use the microbes surrounding the root systems to defend itself against the invasion of a pathogen? Then I divided my presentation in culture-independent approach and culture-dependent approach. Metataxonomics or amplicon studies. There are some people that call to separate metagenomics from amplicon. They called us metataxonomics, whatever. So we did metataxonomics, metagenomics, and metatranscriptomics. Let's go first to the use of 16S genes, and we used the phylotype. And the first thing we did was to compare all these different treatments suppressive, conducive, conducive plus suppressive soil, and suppressive soil heat treated with temperature. And then we compare these different phenotypes to see which microbes are present in there. So the first obvious question was, let's look at which microbes are present in the suppressive soil and absent in the conducive soil, and then we know which are them, and then we target them. But it's not so easy. So when we count the number of species or taxa, or the richness of all these different treatments, there is no difference. The same guys are across all treatments. But the relative abundance of them, it's different. This is what we have here in this dendrogram. So considering these 6 treatments and 4 replicates, all replicates cluster together. And then we see here, this is the suppressive soil, this is the conducive soil. And what it's really interesting here, it's only the presence of the fungus. This is the treatment of suppressive soil without the pathogen and with the pathogen. So only the presence of the pathogen was able to distinguish or to a level of resolution to change the bacterial community in these 2 treatments. And how the fungus does that, it's a really important question for us. So how to select a target group to explore? And then looking at all these treatments, we selected 3 criteria. The first criteria is, first, let's see which microbes are more abundant in suppressive soil than in conducive soil. This is the first criteria, and these are the top dynamic bacterial taxa present in suppressive soil. The second criterion was, when the pathogen is infecting the root system, which bacteria taxa increase in abundance in the presence of the fungus. And the third one, using the transferring treatment, when we add suppressive soil into conducive soil, which microbes increase in abundance and are able to give some level of disease resistance to the plant. So taking into account all these 3 criteria, we end up with this group here, Protobacteria and Firmicutes. So the conclusion here is that based on this study, we saw that gamma and beta protobacteria from these families and also Firmicutes were identified as the dynamic taxa associated to disease suppression. So using amplicons, we could see which bacteria is associated, is strongly correlated with disease suppression. Then we moved to metagenomics, and then we did shotgun metagenomics to look up to which functions are upregulated in the presence of the fungus. So here we selected only 2 treatments, suppressive soil with and without the pathogen. So in the presence of the pathogen, oxidative stress, bacterial alarmone, PPGPP, and secretion system are upregulated when the pathogenic fungus is present. I will come to these functions later with the metatranscriptomics data. But when you do that, we know that these genes, these functions are there, but we don't know if they're really being expressed when the fungus is there. That's why we have to move to RNA to see which genes are being expressed. And then I added here a graph showing data from metagenomics and metatranscriptomics. I'm not sure if I should put both data in the same graph, but the point I'd like to make clear here is that when you look at the shotgun sequencing, we can separate both treatments. This is the suppressive soil without, and the black triangles are the suppressive soil with Rhizoc. We can discriminate the treatments. the 3 replicates. But when you try to do the same looking at the RNA seq, it's difficult. The samples are scattered around the graph. And this is expected because we are looking genes being expressed in a given time point. And we don't know plants coming from different pots and different replicates. They are not synchronized. So this is a challenge, how to use statistics to support the conclusions that when we look at metatranscriptomics data. Yeah.
So what is the underlying data here? What are you plotting? I mean, what are you comparing?
Yeah.
Are you comparing reads or function or abundance in this plot?
Yeah, this is only function.
Okay.
Only function. From shotgun, which functions are distributed in all samples, and the RNA data functions, only functions. Because we want to pick up which functions are more expressed in one condition comparing with the other condition. But even with these limitations, when we did the metatranscriptomics, so I will first tell about the structure of the bacterial community, because as we can take the rRNA, we can look at the bacterial composition of this community, and the groups, the top dynamic bacterial groups revealed by metatranscriptomics were the same when you used phylotype to look at the data. And the question here was that if the fungus is invading the rhizosphere microbiome, is this fungus changing the structure of the community? And we could detect substantial shifts in fungal, viral, and bacterial community. And the dynamic bacterial families comprise members with no antifungal activities, such as Burkholzeriaceae and Oxalobacteriaceae, which are the same families detected when we used amplicons. This second graph is looking at the fungal community composition. Just to illustrate, the top one is the anamorph of Rhizoctonia solani. It looks obvious that in one treatment we inoculate the pathogen and then we are able to identify.
The error bars are replication between samples.
Yeah, 3 replicates. But looking at the functions, we could detect only few functions with statistical support. But among them, we found the same 3 that we detected using shotgun sequencing. Oxidative stress, bacterial alarmon, PGPP, and secretion are upregulated during pathogenic fungal invasion, matching with the metagenomic data. It's a way to support our data. And when we look to this, we don't know exactly the meaning of all these functions and what is going on the rhizosphere, but makes sense. Bacterial alarmon PPGPP, it's known to perceive stress and to coordinate gene expressions in the bacterium genome to face the stress and try to solve what is going on. Secretion, when you look which groups are expressing this function, we have a lot of bacteria well known to produce lytic enzymes to degrade, for example, the wall of the fungus to fight against the fungal invasion. But based on the information that we got here, we would like to demonstrate or to show or to prove that the groups that were pointed by this approach are really working as we expected. And then we move to the last section of my talk, isolation and bioassays. the first thing we did was to isolate, and then we target Pseudomonas. We used a selective medium to target this group, as was pointed by the Amplicon study. Then we isolate Pseudomonas from suppressive soil. We sequence the 16S gene, and then we trace it back, these genes, in the phylotype to see if the same groups, the groups that we are able to take from soil, are the same groups which were more abundant in suppressive soil than in conducive soil. And this was what happened. Beneficial Pseudomonas were more frequently isolated from suppressive soil than from conducive soil, matching with the metataxonomic data. So now we have in hands many isolates. We screened them based on the ability to antagonize the pathogen in vitro. We just grow our isolate against the rhizoc to see if they can inhibit the growth in vitro, and then we select those isolates. And for some of them, we did random transposon mutagenesis to identify which compound is playing a role in disease suppression. This is a simple experiment where we sow the seeds of sugar beet in a tray with conducive soil, and then we add the fungus here. In a conducive soil, the fungus is able to go through the soil, killing the plants. This is the control, and this is the disease progress. When we add to the conducive soil the isolate, the bacterium isolate that were able to inhibit the fungal growth in vitro, we get disease control here. And the last bar is exactly this isolate, CHC52, a mutant of this isolate in which this one had lost the ability to control the disease in the bioassay. So one of the mutant strains of C52, named 033, this one, lost the ability to inhibit the pathogen growth in vitro or in the bioassays. And then moving on with genetics and chemistry, we are able to, as we have the mutants and we use a transposon, we are able to map where was the insertion of the transposon with shut down a specific gene. And then doing sequencing, we came up with this set of genes. We named them thanamycin, and then we have Ta-A, Ta-B, and Ta-C, C12, and Ta-D. We predicted the polypeptide coming from this gene using the conventional code. No problems with stop codons here. But at this stage, what happened is that we would like to really elucidate this compound, the molecule. We struggled for several weeks, months, trying to extract this compound in the culture. We failed to do that, but get in contact with a group of Peter Dorenstein from University of California at San Diego. He was at the same time developing a really nice system in which we just add the plate to a mass spec where a needle released a droplet of solvent in the bacterial colony, and another needle capture the solvent, and this mechanism is connected with the mass spec and connected with a server, with a database, and in 2 days he gave us the structure of this molecule, which is called thanamycin. we also explored this isolate for other bioactive compounds. But in the context of our story, this strain, C52, protects the sugar beet seedlings from fungal infection through the production of the bioactive compound called tannamycin. So then, going back to our system here, I will tell you what we know so far. So based on the Amplicon study, metagenomics and metatranscriptomics, we know that these bacterial groups are correlated with disease suppression. Actinobacteria, protobacteria, these bacterial families, they show strong correlation with disease suppression in soil. These three functions plus tannamycin production are also correlated with disease suppression. So what we think that it's going on here is that when the fungus is invading the rhizosphere region, the plant exudates are able to somehow communicate with those bacteria and trigger these functions to protect the plant against the fungal infection. There are many unknowns here. We don't know if, I have tried to do some experiments to address these issues, but for example, we don't know if the fungus needs first to infect the plant before all signaling take place to tell which bacteria should grow and produce a given compound. We don't know. We don't know if just the fungal germination is enough to tell the microbiome, I'm here, you can fight against me. We don't know nothing about that. I'd like to stop here and thank Jos Heimakers from NIU in the Netherlands, Gary Anderson, Peter Bakker from Utrecht University, and Peter Dorenstein. And just before the questions, I'd like to tell you what we are trying to do with this knowledge. Where are we gonna go with that? So if this is true, if the plant genes are able to shape the bacterial or the microbiome in the plant roots, and the plant relies in some specific traits of this community, we can take advantage of that to try to solve many problems. For example, if you can manipulate the plant genome to overexpress these genes able to talk to the microbiome, we can use this strategy and then reduce the amount of chemicals used in agriculture. And if this is really true, there is another hypothesis that we are exploring now, is that if the plant co-evolved with this microbiome together in nature, the hypothesis is that plants growing in native habitats are much more interacting with the plants than modern cultivars that we have today. And we can imagine that during the breeding process, those genes able to shape the plant microbiome were lost over the breeding process. Because during breeding, we supplied nutrients, we used chemicals, the plants doesn't have the same pressure that have in native habitats. And then what we wanna do is to go to investigate the wild types, wild plants in native habitats, and check if these plants are able to recruit actively more beneficial microbes than a modern cultivar. And if this is possible, then we're gonna use metagenomics, metatranscriptomics to search correlation between the microbial community structure and the plant background to try to map which genes in the plant are able to be used in a breeding program nowadays. That's it. Thank you for your attention.
Thank you very much.
Welcome.
Any questions? Yeah, please.
Do you think it's probable that other suppressive soils in other places will have communities maybe that still inhibit rhizoctonia on sugar beet but produce completely different antibiotics?
Yeah, I think this is a very specific case. So the fact that we found this tannamycin compound, we were not able to find these genes looking at the metatranscriptomic genes. So looking at the rhizosphere, what I think that it's going on there is that when the fungus is invading the plant, any other antibiotic able to play a role in this process will be activated firstly. We just get, by chance, we end up with this one. But I can imagine that a set of numerous different antibiotics are playing the role at the same time.
Another question. You had a question?
Yeah, that maybe sort of partially answers my question, but I was just going to say, so you found the tannamycin gene in this cultivated Pseudomonas, right? Did you then go and look for that in other organisms in your metagenome data set?
Yeah, we couldn't find back, no, because sometimes this is a really powerful antibiotic, expressed in really small amounts, but with a big effect. And then the resolution that we got doing metatranscriptomics was not able to reach the abundance of this gene.
I was asking about the metagenomics rather than the meta.
Yeah, we also didn't find. Yes.
So you kind of assume that the plant is playing an active role in this process. But I mean, a kind of simpler way to do it is that when the fungus comes, that there are certain bacteria that can thrive by attacking the fungus, and they just multiply. And in some sense, it just seems that the number of those bacteria multiply, isn't that? I mean, you find the genome, you find the metagenome is different.
I see.
So in some sense, maybe the plant is just sitting there, and it's just a war going on between the bacteria and the fungi.
Yeah, but the way that this suppressive soil is shaped in agriculture, they are observed normally just after a disease outbreak, and then we keep cultivating the same plant in the same soil for 4 or 5 cycles or years in a row, and then you're able to have a field able to suppress a disease. So the condition is to have the pathogen and to have susceptible hosts in the soil.
Okay, but I mean, so if the pathogens are absent, if the bacteria that eat the fungi are absent in regular soil, and by doing that for 4 seasons, you're just slowly amplifying up the number of the bacteria.
Yeah, but then we don't have a suppressive soil. If you test the soil without the plant, and then you're gonna get diseased plants. The soil, the microbiome is not able to protect the plant. So if you remove the plant from the figure, you keep adding the pathogen in the soil.
Okay.
In one treatment. In another treatment, you use the plants. And after 4 years, when you do the same, the soil without the plant is not able to protect the plant. And in this, there is some sort of pressure, selection pressure in the community, able to select those bacterial groups able to protect the plant.
Another question there.
So what happens if you used a different plant? For those 4 years.
It's a different story.
Instead of fallowing, you actually use a different plant. Do you then maintain the population or not?
Yeah, it's a different story. We have been working with 3 different systems. This is sugar beet and rhizoc, also wheat and guaiacamomyces, and also flax and fusarium. And we tried to change the players. And this seems to be a very specific interaction between a given pathogen, the host, and the soil.
Is there any evidence that the microbes themselves, which are beneficial, are actually, the plant is actually doing something to make life good for them, so that they're hanging around. So I'm not sure if you covered that, but you have evidence for that.
This is a two-way contribution, because the amount of carbon exudates released by the plants and consumed by the microbiome is huge. And then the microbes are attracted by those exudates and they consume what is released in the soil. And then firstly seems to be that the bacteria is taking advantage of the plant exudates. But after that...
Yeah, but this would also apply to the harmful microbes. So, I mean, is the plant doing something to differentiate between the good guys and the bad guys?
Yeah, we don't know. What we know, I didn't mention here, is that when you have conducive soil, the abundance of potential human pathogens in the rhizosphere is higher than when we compare with suppressive soil. Apparently, after let's say the plant is growing and has an established microbiome, and if something disturbs the equilibrium, and then the disequilibrium goes everywhere, and potential human pathogens also increase in abundance in this system. But we don't know how the plants are able to differentiate that.
Are you using the next generation sequencing to analyze the metagenomics?
Yes, Illumina.
Yeah, Illumina.
Illumina.
Did you find some of the reads cannot be mapped or unknown? How many percent?
Yeah, we have been using the MGRAST pipeline for everything. So then we just go through the MGRAST pipeline. But a portion of these genes are excluded from the analysis.
Because I wonder whether there's some species, which not known.
That's a really important question, because of course we are showing the structure of this microbial community, but there is a bias in the database which comes from the ability that we have to grow microbes. So the target group that we used here are very well known group able to be cultivated, and this was also another reason why we're gonna go for them, because we know how to deal with them, how to grow them, how to do mutants, and then we can use them as a proof of concept. But a big portion of this data is really unknown. We don't know. We don't know.
What percentage?
The person.
Okay.
Yeah, I would say about 25% in our data. Yeah.
Take one more question, because I don't...
that was a lovely story. What do you know about the natural product? Is this, its structure? Do other organisms have it?
The tanomycin?
Yeah.
No, it's a new one. It's a new one. It resembles some other antibiotics, but it's a completely new one.
Right.
Yeah, and then when
produced it, you haven't made it.
No, not yet.
Characterization from mass spec.
Yeah, it's we just have that, this live mass spec technology. Yeah, we are not able to extract or to produce this compound, because then we would use in our system to see if this compound is really fighting against the fungus or if signaling something there. Yeah, we don't know.
Okay, well,
Okay,
thank you
thank
very
you.
much.
Thank you.